All right, welcome back. We're going to continue with fermentation, section 8.4. So in this section, we'll look at why fermentation does not require oxygen, look at the different types of fermentation pathways and their unique products, and then finally compare and contrast fermentation with anaerobic respiration. When respiration is not possible, either because oxygen or an electron transport chain is missing, then cells will use fermentation. And fermentation uses an organic molecule, like pyruvate, as the final electron acceptor. It regenerates NAD+, so that glycolysis can continue, but only makes 2 ATP per glucose. This table from the book compares respiration to fermentation in terms of the number of ATP that are formed. So in aerobic respiration, in the previous video, we learned this is the maximum, or this will yield the maximum number of ATP. In anaerobic respiration, we learned that this will yield fewer ATP, and the number depends on the specific organism. The reason we have fewer in anaerobic respiration is because, one, there is lower electron acceptor efficiency, because these electron transport chain proteins will have lower redox potentials. They have lower affinity or attraction to electrons, which results in less energy overall. They also have fewer proton pumps used because many of these anaerobic respiration protein chains will have fewer carriers. So their electron transport chains are physically shorter or fewer. Fewer proton pumps means fewer protons crossing the membrane and fewer protons available for that proton motive force. Finally, fermentation is the lowest with only 2 ATP generated, and those ATP were the ones generated during glycolysis. There are many types of fermentation depending on the products generated. So one example here is lactic acid fermentation. We also have alcohol fermentation, mixed acid fermentation, and many others. Let's start with lactic acid fermentation. In this one, the product of lactic acid fermentation is lactic acid, and it's used by bacteria like lactobacillus and streptococcus, and even in our own human muscles when oxygen is low. So in humans, we have many lactic acid bacteria inside our bodies, such as in the vagina and the gastrointestinal tract. In terms of products, we often use lactic acid bacteria to produce foods such as yogurt, cheese, sauerkraut, or even kimchi. And here's an example of the chemical reaction for lactic acid fermentation. Pyruvate, I'll just look at the bottom one here, it's the same thing. Pyruvate is going to be converted to lactate. Lactate is the same thing as lactic acid. And during this reaction, we can see NAD+ is formed from NADH. This is an oxidation reaction, right? I'm losing electrons here to form NAD+. That means, since they go hand in hand, this must be a reduction. So here is a redox reaction. And the purpose of NAD+ generation is that we need it in glycolysis. Another type of fermentation is alcohol fermentation. So in alcohol fermentation, which is carried out by yeast, Saccharomyces cerevisiae is a very common example. Pyruvate is going to be converted, pyruvate is also called pyruvic acid, is going to be converted into ethanol and CO2. And this process makes beer, wine, and bread. And if I look very carefully at the reactions, the overall reaction is pyruvate turning into ethanol plus CO2, but the detailed reaction involves multiple steps. So first, there's an enzyme known as pyruvate decarboxylase that's going to turn pyruvate into CO2 and acetaldehyde. Then a second enzyme, alcohol dehydrogenase, turns that acetaldehyde into ethanol. So acetaldehyde kind of cancels out here because it's the product and the reactant in another reaction. So the final reaction shows pyruvate as the reactant and ethanol and CO2 as our products. And again, I'm regenerating NAD+, so it's available for glycolysis to occur. And we also have other pathways of fermentation that produce different end products, like propionic acid in Swiss cheese, or acetone and butanol in some industrial solvents. Microbiologists can actually identify bacteria based on their fermentation products. So we're going to do that in lab. Fermentation products can be used to ID what we have, identify the bacteria we have. For instance, in detecting CO2 production or acetone production during butanediol fermentation. These are examples of labs that are coming up for our metabolic unknown. Even the ability to ferment another carbohydrate known as mannitol can be used to differentiate mannitol fermenting Staphylococcus aureus from other non-mannitol fermenting staphylococci. Overall, all of these pathways of fermentation ensure that we have enough NAD+ so that glycolysis can continue and we can make those 2 ATP per glucose. And you do not need to memorize this table, but it's a nice table that illustrates many of the common fermentation pathways. We saw earlier the lactic acid pathway, the alcohol fermentation pathway, and then there are several others that are usually specific to certain species of bacteria or yeast or other microbes. And then you can see many of the common commercial products that are produced through fermentation. And if we compare respiration versus fermentation, respiration uses an electron transport system or chain and has a much greater ATP yield. Remember that aerobic respiration can generate up to 38 ATP per glucose. In fermentation, there is no electron transport chain, no electron transport system. You only generate 2 ATP per glucose. Let me write that. That's per glucose. So both in the end do help us regenerate NAD+ because that is a reactant necessary for glycolysis. So when would a metabolically versatile microbe perform fermentation rather than cellular respiration? So a microbe capable of both will switch to fermentation when 1. Oxygen is absent, so it cannot perform aerobic respiration, and 2. When there is no suitable inorganic electron acceptor available for anaerobic respiration. For instance, there's no nitrate, no sulfate available. And fermentation will allow the cell to regenerate NAD+ so that glycolysis can continue, even though the amount of ATP you're going to get is going to be much lower, only 2 per glucose. So one good example we'll see in the lab is E. coli. E. coli will go through respiration when oxygen or nitrate is available, so oxygen for aerobic, nitrate for anaerobic respiration, but it can also ferment sugars when neither is present. Fermentation pathways are used to identify microbes in the lab. They're also the basis for important products, foods like cheese, wine, biofuels, and even probiotics. So this makes fermentation both medically and commercially significant. Scientists have also developed techniques that identify bacteria according to their biochemical characteristics, such as their fermentation products or other metabolic reactions. And in many tests, microbiologists use individual test tubes and plates to do or perform the biochemical testing, and we're going to do the same thing in lab. However, when time is critical and what's happening in many clinical labs today, we see multi-test panels like the one shown here below. These contain a number of miniature reaction tubes, so all of these are miniature reaction tubes. Each usually can include some kind of specific substrate and pH indicator, and after you inoculate the test panel with a small sample of the microbe, so you would inoculate all of these panels with a little bit of the microbe, we can incubate it for a day or two and then compare the results to a database that includes the expected results for specific biochemical reactions for a bunch of different known microbes, and that will enable very rapid identification of your unknown microbe. So test panels really allow us to reduce costs while improving efficiency and reproducibility by performing a large number of tests at the same time. We're going to be doing something similar in the lab as well during our study of GI tract bacteria. All right, let's recap 8.4 fermentation. So the first one, define fermentation. Why doesn't it need oxygen? Fermentation is a metabolic process that regenerates NAD+ from NADH by using an organic molecule like pyruvate as its final electron acceptor. It produces ATP only through glycolysis, so you only make 2 ATP per glucose, and it doesn't require oxygen because it completely bypasses that electron transport chain. For the second one, fermentation pathways and their end products and examples. So we have lactic acid fermentation. The product is lactic acid, and microbes like lactobacillus or streptococcus undergo lactic acid fermentation. We also have this in our muscle cells under low oxygen conditions. Uses could be to make yogurt, cheese, or sauerkraut or kimchi. For alcohol fermentation, our next type, our end products are ethanol and CO2, and a microbe example would be Saccharomyces cerevisiae, the yeast, and uses could be to make beer, wine, or bread. There are also many other microbial pathways of fermentation. There's mixed acid fermentation, butanediol, propionic acid to make Swiss cheese, so just know that many others exist. Looking at the last one, comparing and contrasting fermentation and anaerobic respiration. So fermentation, there's no electron transport chain. The final electron acceptor is an organic molecule like pyruvate, and there's very low ATP created, only 2 per glucose. In anaerobic respiration, it does use an electron transport chain. So the final electron acceptor is going to be some kind of inorganic molecule, something that's not oxygen, like nitrate or sulfate. There's a greater number of ATP generated compared to fermentation, but it's still lower than aerobic respiration. So key similarity is they both occur without oxygen, but a key difference is that fermentation is electron transport chain independent. There's no electron transport chain, while anaerobic respiration still relies on that electron transport chain. So in the previous section, we talked about the catabolism of carbohydrates. Now we're going to switch gears and look at the breakdown of lipids and proteins. We'll look at the enzymes and products generated and see how we can use these enzymes and products to identify unknown bacteria. So lipids, including triglycerides and phospholipids, are broken down by enzymes called lipases and phospholipases. The glycerol backbone of these lipids is converted into glyceraldehyde 3-phosphate, or G3P, and that will enter glycolysis. Fatty acids can go under a process known as beta oxidation, where you're breaking down the chemical bonds found in those long fatty acid tails, and they can produce acetyl-CoA through this process. The acetyl-CoA molecules feed into the Krebs cycle. This overall process, or these two processes, will generate large amounts of NAD and FADH2, and these molecules will fuel ATP synthesis through oxidative phosphorylation. Clinically, some of these microbes can use lipases and phospholipases as virulence factors to cause disease. For example, Staphylococcus aureus can destroy our host cell lipids, and some other pathogens like Mycobacterium tuberculosis can even degrade cholesterol to survive inside the host. So this might be the cholesterol that is in our cell membranes. So these enzymes can contribute to virulence in microbes, allowing them to cause disease in the body. Lipases and phospholipases break down host lipids and membranes. So this allows microbes to invade tissues, escape host defenses, and access nutrients at the same time. Staphylococcus aureus, for instance, uses these enzymes to damage host cell membranes. And if this were to happen in our immune cells, like the phagocytic cells, macrophages, or neutrophils, then the lipids in their cell membranes would be broken down, killing or damaging these cells, and the bacteria can thereby avoid being engulfed or destroyed by these immune cells. Proteins can also be broken down by microbes for energy. Proteins are first broken down into peptides by enzymes known as extracellular proteases. These are then further broken down into amino acids. Amino acids are then deaminated. That means you're going to remove the amino group from the amino acid, and their remaining carbon skeletons can enter either glycolysis, that transition step, or the Krebs cycle as intermediates. Clinically, protein catabolism is useful for diagnostics. We're going to perform several of these in the lab as well. The gelatinase test, for instance, can detect organisms like Proteus and Serratia, where they have an enzyme gelatinase that can break down gelatin protein. And the caseinase test can identify species like Pseudomonas aeruginosa, where they have an enzyme to break down casein milk protein. So protein catabolism can help us identify microbes. Many microbes secrete enzymes called proteases that break down proteins into their smaller peptides or amino acid monomers. And specific enzymes can be detected with lab tests that we will be performing. The gelatinase test I mentioned can help identify Proteus and Serratia that are positive that have this enzyme. The caseinase test can be used to identify the presence of Pseudomonas aeruginosa, and positive or negative test results will really help us identify and differentiate species in the lab. All right, let's recap the lipid and protein catabolism section. So the first one, lipases and phospholipases break lipids down into glycerol and fatty acid components. Glycerol can enter glycolysis as glyceraldehyde 3-phosphate or G3P, whereas fatty acids will undergo beta oxidation, producing acetyl-CoA that can enter Krebs cycle. And both of these help to generate lots of NADH and FADH2 that can eventually produce ATP through oxidative phosphorylation in that electron transport chain step. For the second one, describe how lipid catabolism can be used to identify microbes. We know that some pathogens produce lipases and phospholipases that break down host cell membranes, and their activity can be detected in the lab. So we do have several lipid hydrolysis tests, and the presence of these enzymes can indicate virulence as well, the ability to cause disease such as in S. aureus. For the third one, how proteins are catabolized. So proteases will break proteins down into peptides and then the monomers, which are the amino acids. Amino acids are then deaminated, where the amino group is removed. And these intermediates can enter glycolysis, the transition step, or the Krebs cycle. So this allows microbes to use proteins as both energy and carbon sources. In the next one, the very last one, we're going to look at how protein catabolism can be used to identify bacteria. So bacteria differ in their ability to produce very specific proteases. We talked about the gelatinase test. So this is going to show us that these bacteria can break down gelatin and it'll go from a solid to a liquid state. And we'll see this in the lab portion. For the caseinase test, we're going to see that bacteria like Pseudomonas aeruginosa can break down casein milk protein, turning it from an opaque liquid to a clear liquid. And these positive or negative results can help us differentiate different bacterial species. And now we'll switch gears again and go to the next section. 8.6 covers photosynthesis. We're going to look at the function and locations of different photosynthetic pigments in both eukaryotes and prokaryotes. We'll look at reactions known as light-dependent and light-independent reactions. Look at the reactions that produce glucose and compare and contrast cyclic and non-cyclic photophosphorylation. Photosynthesis converts solar energy into chemical energy, and it occurs in plants, algae, and many microbes. There are two stages in photosynthesis: the light-dependent reactions and the light-independent reactions, also sometimes called the Calvin cycle. The light-dependent reactions are going to capture energy from sunlight and use that to make ATP and NADPH, another type of high-energy electron carrier. These products are then going to power the Calvin cycle, in which we're going to see carbon is fixed into organic sugars. So I like this picture because it shows both the light-dependent reactions and the light-independent reactions where carbon fixation is occurring, also called the Calvin cycle. So here on the left, we have those light-dependent reactions. We're going to see a series of steps occur to produce lots of ATP and NADPH for us. Those products are going to power carbon fixation, taking inorganic CO2 from the atmosphere and producing organic carbon molecules such as glucose as products. In eukaryotes like plants, photosynthesis happens in these organelles known as chloroplasts. The light reactions will occur in the thylakoid membranes that are organized into stacks called grana. Singular would be granum. The Calvin cycle, or the dark reactions, are going to happen in the stroma, the liquid portion inside the chloroplast. Prokaryotes, though, like bacteria, do not have membrane-bound organelles like chloroplasts, so instead they're going to use infoldings of their plasma membrane that contain pigments. So these are going to be thylakoid-like structures that contain photosynthetic pigments. Different pigments absorb light at different wavelengths. Chlorophylls, for instance, that are found in plants and certain photosynthetic bacteria, appear green, but they actually do not absorb green light. Chlorophyll pigments absorb red and blue light. And if you look at this absorption spectrum here of different pigments, here's chlorophyll, different forms of chlorophyll. These absorb in the blue region, and they absorb in the red region, but they do not absorb green, and therefore they reflect or transmit green, and that is what we see. Carotenoids are another type of pigment. They're going to appear reddish-orange. They appear reddish-orange because they don't absorb in that region. Carotenoids absorb green or blue light. Pigments are going to be arranged in photosystems in our photosynthetic organisms, and these photosystems are composed of an antenna complex and a reaction center. We're going to see that light excites electrons in the reaction center, and these electrons will be then passed on to an electron transport system to make ATP and NADPH. So here's an example of my photosystem. I have a bunch of light harvesting complexes or pigments that will pass the energy from light from one pigment to the next until you get to the reaction center. Once the energy is passed to the reaction center, it is going to become oxidized, where it loses an electron. That electron is passed to an electron transport system, and there will be a proton motive force generated, just like we saw in the electron transport chain previously, and the electron will eventually be used to reduce NADP+ to form NADPH. These two are going to be used to power the Calvin cycle later on. We're also going to see that this reaction center, it lost its electron, so that electron will later need to be replenished. In a phototrophic eukaryote, where does photosynthesis take place? We said it occurs in chloroplasts. So the light-dependent reactions are going to happen in the thylakoid membranes. The light-independent reactions, or the Calvin cycle, are going to happen in the stroma, which is just the fluid portion inside the chloroplast. Earlier, I mentioned that the reaction center lost its electron. Remember that electron was passed to NADP+ to make NADPH. So now we have to eventually replenish that electron. And that is done over here, that's shown over here. And this will determine whether your photosynthesis is oxygenic or anoxygenic. So if the molecule that donates an electron to the reaction center is water, so water is going to donate the electrons, forming oxygen, then this is going to be known as oxygenic photosynthesis. And this happens in plants and cyanobacteria. Anoxygenic photosynthesis is when their donor over here is not water, something else like hydrogen sulfide that does not produce oxygen as an end product. And this will be seen in purple and green bacteria and others. Here's another look. In oxygenic photosynthesis, water will be the one to donate electrons to that reaction center, forming oxygen. In anoxygenic photosynthesis, it's another type of donor, and the product is not going to be oxygen. All right, that takes us to the end of our third video, and we're going to wrap up photosynthesis and finish the rest of chapter 8 in the next video.